Thank you very much for having me here today. So I'm actually rather glad I followed Phil's talk because there's a lot of stuff that he talks about that fits perfectly with some of the things that I will briefly touch on. Um, my aim of the talk really is to just go over the last 6 years of our transplant consortium and just give you a taster of what the next 6 years, hopefully, we find out today, will be for the next 6 years. So in 2017, we first got our Genome Canada grant, which was to fund the Genome Canada Transplant Consortium, which pulls together about 20 institutes and about 80 PIs to look at the ways that we could reduce antibody-mediated rejection. So the principle of our first grant for 6 years of this program was to reduce the frequency of antibody-mediated rejection in renal transplantation. And then the second was to, by doing that, hopefully minimize the demand on the healthcare system here in Canada and use that as a framework to provide to our international collaborators. And then third, to improve the societal care of major chronic diseases, renal transplantation being one, but the knowledge that we gain from the immune responses would also be able to help understand different policies for autoimmunity and oncology. So the way that we structured the Genome Canada Transplant Consortium was a 4-pillar approach. And we had the first pillar is epitope matching. So I will go into that a little bit in more detail in a minute. The second pillar was to look at immune monitoring methods and approaches that we could use to follow the transplants patients post-transplant. The third pillar was to optimize therapeutics, the way that we manage our patients and personalize the management post-transplant. And our fourth pillar was to look at the evidence-based health policy recommendations, including legal and ethical guidelines, communication, and have public and patient and provider perspectives included in some of our feedback to the government and funding agencies. So we wanted to also incorporate the ability to translate some of these new advanced technologies that were coming through the research arena into the clinic so the grant was actually for 4 years, but we got a 2-year COVID extension, so the 6 years that we had. So our first grant was quite specific. They wanted to get these assays into the clinic and impacting patients within the 6-year timeframe. So we had a couple of key technologies that we targeted, deep immunophenotyping using flow cytometry, rapid sequencing, which I'll cover in a little bit, single cell biology / proteomics. I've sort of plunked them into one pillar, but I can explain why in a minute, and antibody and sort of HLA antibody detection. So one of the first technologies that we looked into was the rapid sequencing for high resolution typing of HLA genotypes for patients and donors. And it was quite interesting at the Genome Canada pitch. We actually proposed using this at the time, very exciting, very new technology that could do sequencing in a blink of an eye, in using this tiny sequencer.
Just in 2 minutes.
Tiny sequencer that was about the size of a USB stick, and we were told that would never work. And regardless, we pushed on, and 2 years into our proposal, I was able to sequence Dr. Keon in the time of a lab meeting and show this to the Research Oversight Committee, who were quite blown away and actually said, we should not have doubted your abilities to get this moving. So as part of a Canadian program, we've actually now implemented rapid high-resolution HLA genotyping using the Oxford Nanopore platform to allow us to do epitope matching for our transplant programs. And the reason that this is important is because we believe that by doing eplet matching rather than HLA antigen matching, this reduces the variability that we see in the HLA genes, and allows us actually to prospectively match a donor against a recipient to reduce the immunogenicity and the likelihood of developing rejection down the line. But in order to do that, you have to have high resolution typing for your HLA genes for both the donor and the recipient. So if you have a living donor, that's quite feasible. You have about 4 days in which to type a living donor. But in a deceased donor setting, you only have about 6 to 8 hours to work up that that donor. And so that's why the rapid genotyping became critical in order to allow both our donors, deceased donors and living donor wait lists to be provided with the opportunity to be eplet matched. So one of our recently graduated PhD students, Jenny Tran, right here, actually published on this and published on the B-cell eplet landscape of the British Columbian transplant population, and showed that the reduction in complexity from HLA genes down to eplets. And in fact, we've actually moved forward from that, not only looking at the lay of the land in terms of the amount of epitopes that are present in our donor and recipient populations, but also to see whether or not mismatch level of these eplets does in fact mean that you are more likely to develop de novo DSAs post-transplant, depending on your level of eplet mismatch. So another platform that we had been developing with Dr. Franz Benninger sitting on the front row over there is looking at deep immunophenotyping. And this is to allow us to understand the types of immune cell subsets that are present in the periphery. At the moment, we look mainly at the peripheral blood only. And we've had a panel of a complex panel of 6 subsets, so B, T, NK, macrophages, and monocytes. And we have this very cute BD cytoflex, which is quite tiny considering what flow cytometric machines look like. And Franz has been able to show us in uremic patients in the pre-transplant setting, so in a subset of patients, so where we have samples pre-transplant and then compared to healthy controls, age-matched healthy controls, how they differ in their immune subsets between uremic patients and healthy controls. So we've just highlighted in this Tisney plot here where you can see some of the subsets that are colored. I don't know if you can see the legend, but some of the NKT cells are significantly reduced in our pre-transplant patients versus our control patients, as well as some of the NK CD15 positive cells at the bottom there. And then we've taken this a little bit further and looked at the same patients longitudinally over the first 90 days, which is the sort of critical period of trying to make sure that these patients don't have any early onset rejection. And so accounting for their immunosuppressive conditioning at the time of transplant and post-transplant, you can see that some of these subsets actually return back to come back to sort of normal baseline levels, almost normal baseline levels over the first 90 days, and how this changes from day 0 up to day 90. So this just shows you the power of using deep immunophenotyping and looking at the cytometric subsets in these patient cohorts, both at the time of transplant to identify patients that might be at more an increased risk of developing rejection, and also following them as a monitoring tool post-transplant. Another assay that we had looked into but have not been able to get into clinic is looking at immune cell metabolomics. No, I always get these mixed up. Metabolites. No, metabolism. I always get them mixed up. So this is where we look at the cells and the way that they basically breathe and use their mitochondria to produce ATP, and that we look at 2 different pathways: the glycolytic pathway and the oxygen consumption rate. So we use a platform called the Agilent Seahorse platform, and this is a sort of a well that looks at you lay out your cells across the well, and it's able to measure within those cells over the space of a couple of hours how what the capacity of those cells are for respiration. And so the hypothesis is that these bioenergetic profiles of lymphocytes that are either healthy or perhaps sick in the uremic setting or immunosuppressed in the post-transplant setting can give us an idea of whether or not these lymphocytes are able to mount an immune response. So, tired cells may have a lower capacity to actually respond to an antigen, whereas healthy cells that are not immunosuppressed may have more of a capacity to respond to an antigen. Another assay that we've been looking at, and hopefully we'll get some quite interesting data very soon, is looking at immune cell repertoire profiling. We focused initially at T cell repertoire sequencing, and this is done with our partner Adaptive, and we look at the T cell receptor because this is specific for an antigen. So each T cell clone is specific for a particular antigen in the context of transplantation. We would hope that we would see specific T cell clones, and therefore the receptor that sequences specific for that antigen for the donor antigens that are mismatched between the host and the recipient. The way that we decided to utilize this immune cell repertoire profiling is as a monitoring tool, actually, not just to identify the particular clone. So pre-transplant, we would have a MLC, so we'd mix donor and recipient cells together, and the recipient cells that were able to recognize and mount an immune response and proliferate in the Petri dish, we would then take everything and sequence that, and the proliferated T cell with its single specific T cell receptor would be the biomarker that we would identify in the T cell sequencing data. So we would use that marker and then take that marker as specific for that patient moving forward into the post-transplant space. We would take longitudinal samples as a monitor, just blood samples is sufficient over time. And as long as we don't see an expansion of that specific clone that was identified in the mixed lymphocyte culture pre-transplant, we would assume that there was no activation of your donor-specific T cells in the patient. So this is to track and as a post-transplant monitoring to give us more of an idea of the whole system response to the donor graft. We've also recently started moving into proteomics and more advanced antibody detection. So we have been working with a company called Olink, who I think are here today, and we're looking at proteomic profiling of pre- and post-transplant patients to understand the immune mechanisms involved in, well, in this case, rejection, but we're also, we want to know the balance between rejection and tolerance. The way that we've been doing this right now is look at the cytokine profiles in patients that have a pre-transplant and how that changes post-transplant, taking into account that, of course, these patients will be immunosuppressed, and what are the differences between patients that reject versus those that don't reject. We also monitor antibodies. Clinically, we have an assay called the Luminex assay. That assay has been around for some time and is potentially problematic in interpretation. And so we've been working with another company called Fluidics that may have a more high resolution method that doesn't require surrogate detection of the actual anti-HLA antibodies in the patient's serum. And of course, the use of the reason that we want to look at all of these things is to build a sort of portfolio of biomarkers, cytokines, antibodies, TCR repertoire that can be included into this polyomic score of immune risk. And then we could say to the patient, okay, looking at these scores that you have, you are at this risk, intermediate, high risk, low risk of, early, late, or whatever, rejection. That would allow the patient to have sort of more comprehensive understanding of the prognosis of their transplant. So some of the other stuff that we've also been doing is to look at transcriptomics, and Phil kindly presented some of this already. We've actually done I'm going to jump straight to the next slide because my data's on that. Done some very, very similar studies to what Phil has done. We had a cohort of about 500 transplant patients, and we used, this is back in the day when bulk sequencing was, gene chip sequencing was all the rage. And so we were using Affymetrix gene chips at the time and looked at the pre-transplant versus post-transplant profiles of these patients. And actually, we've also published in a very similar manner, I think it's a 15 transcript set that would allow us to discriminate between patients that were at increased risk of developing rejection post-transplant. And then we're transitioning some of the data that we ran in 2015, 2016 from the gene chips into the single cell sequencing arena, where we're working with the group 10X, I'm sure many of you know, to look at sequencing these transcripts and identifying specifically the cell subsets that are involved that are driving that signal that you see in the bulk transcriptomics methods. Another thing that we also have developed in the lab and hopefully will be implementing within the next 12 months for the clinical programs is to use donor-derived cell-free DNA. Cell-free DNA has been proposed to be sort of a liquid biopsy, although with our work, we realized that perhaps this is not so much a liquid biopsy, but also another metric that we can use and combine with our polyomics risk score to provide a clearer and more high-resolution picture of what is happening within the graft and also within the patient. This can be done using digital droplet PCR. The turnaround time is quite low for that, about a couple of hours, which means it is quite a useful tool. It's much faster turnaround time than a biopsy, although it doesn't give you the same resolution as a biopsy. So it just is, perhaps a flag to tell you, yes, we actually do want to move ahead with the biopsy, or maybe we don't want to move ahead with the biopsy at this point. I will maybe just mention this very briefly. We also looked at complications that are due to being on immunosuppressants for a long period of time. The main ones being virology, CMV reactivation, and PTLD. These are two aspects that I'll cover maybe a little bit more at the end of my talk when we talk about the next 6 years. I'll maybe jump over that. I'm being flagged for my time. And then pillar 4, we cover the social, economical, legal, and ethical implications. And the way that this was done was driven by a team that's led by Dr. Sterling Bryan out at UBC. And they built a team that helped plan and steer some of these policy conversations and the deliberative democracy conversations that helped produce actually a really wonderful information booklet that we can now provide to our patients to help them understand the changes that we're proposing with our Genome Canada Consortium, eplet matching and all that kind of stuff. And we actually produced an animated video as well, which if we're lucky, we can see maybe later today. Some social implications. We've also looked at how social media impacts the way that donors come forward, living donors have been coming forward on Facebook and the implications and ethical implications of that. And some of these findings will now be moved into clinical trials, especially now we're going to be moving towards prospective national matching in a national program that's being coordinated by CBS, the Canadian Blood Services. We'll be using real-time data management and using an adaptive trial design with O'Brien Fleming stopping rules, so that this is actually taken from oncology. Oncology has been doing this for many years, and it allows us to actually see the outcome of our changes being made much faster than traditional clinical trials. So I'm going to skip that. I'm running out of time. I just wanted to say that the next 6 years is an evolution of the Genome Canada Transplant Consortium. We want to move away from looking at rejection because the last 6 years has actually shown us that antibody-mediated rejection may not be the critical piece that will impact most of our patients. What we would like to do is start to take steps towards operational tolerance. Operational tolerance is not new. It has been around for many, many years, 20 so years, but it's still sort of the future of transplantation. And so what we're hoping to do with the next 6 years is to take everything that we've developed in the previous 6 years and refine that and help take that towards getting closer towards operational tolerance in our transplant patients. So the way that we did that, again, was a very similar sort of 4-objective approach. Objective 1 was to optimize ex vivo organ preservation and use immune evasion techniques to sort of hide the graft by using bioengineering and other methods to immune cloak the incoming transplanted tissue. Objective 2 was to try to induce operational tolerance through better matching by and also by using modified stem cell infusions. And then objective 3 was to implement immune surveillance, sort of using some of the techniques that we've developed in the past 6 years to monitor the health of the graft and immune status of both the host and to identify risks of complications like infection and malignancies post-transplant. And then objective 4 is to establish the clinical, societal, and health system benefits of these applications. And so this group is actually a much larger group. We've pulled together from an international sort of conglomerate of about 200 PIs in nearly every continent, not quite, Phil being one of our collaborators. And so pillar 1, as I mentioned, was look at transforming the way that ex vivo storage for solid organ is currently happening. And we're working with a group in Western University. Alp Senna has developed this kidney, sort of looks like a kidney in a box, but it involves different temperatures and fluids that flow through the kidney to reduce the amount of injury that happens to the tissue during the transplantation surgery. We're also working with a company called Bryonics, who have developed this solution that you can soak your organs in called immunocloak, and it sort of gives you 28 days to allow the graft. It sort of hides the graft from the host immune system for about 28 days, and then hopefully allowing you the graft to accommodate into the host body within that time. The next phase was to induce operational tolerance using stem cell transplants and in the context of solid organ transplants. And there have been a number. This is figures taken from a great review article that just covers all the kinds of immune subsets that have been found to have regulatory impact on tolerance. None of them are the magic bullet, but potentially by tweaking them in an appropriate manner and in a personalized manner, that we might be able to get these patients closer towards tolerance, both in the stem cell setting and in the solid organ setting. And also with that, we're working with Peter Zanter. He's one of our lead PIs on the new grant for the next 6 years, as well as Alice Betena from Stanford, both of who work in the stem cell arena. Peter Zanter works in stem cell bioengineering, and he's going to be helping us identify ways to better improve and synthetically modify stem cells, that we could use them either to regenerate some of the solid organ tissue that we're using, or else to actually improve the efficacy of pushing towards a tolerogenic phenotype in our transplant patients. This has already actually been done by Alice Betena using alpha beta T cells and CD19 positive B cell depleted transplants in the context of solid organ at the same time as a solid organ transplantation. We will also be trying to drive these forward with clinical trials. We have collaborators here today that will also be speaking about that in the stem cell session, which is this afternoon. And we're also working with SFU, who has a synergistic drug screening center, who will be helping us to identify ways that we can maybe do synergistic drug screening and identify novel therapeutics. So just to wrap up there, that was a whistle-stop tour of the last 6 years, and hopefully I gave you a taster of what the next 6 years would look like. We've just submitted this roadmap to Nature Reviews Nephrology that will basically outlines our legacy plan. Hopefully we will be successful, and so we won't lose all the momentum we've gained in the previous 6 years with the Genome Canada Transplant Consortium. But this is to extend the success so far and take some of this forward and expand the opportunity for coordinated and personalized and hopefully decentralized care delivery, and to ensure that the societal and scientific and translational success of this partnership is actually shared across all the partners that we have, both in Canada and across the globe. So with that, I'd like to thank all our funding agencies, all our industry partners, and all the people that make up the Genome Canada Consortium, previously and new members that will be joining us hopefully in the next 6 years. Thank you.
Hello. Thank you, Karen, for a great talk. I think we might have some time for one question, if there's any from the audience here. Ahmed, yeah.
Hi, Karin. Thank you so much for this nice talk, and thank you for the introduction, introducing us to this important work that you're doing for the Transplant Society. So my question is just like a basic question. I usually ask this same question to myself. Like, do you think we became more cautious than we should be? Like, are we giving a lot of information to the physician that they don't really need to know? And are we scaring them more? Like I'm talking because in the past, for example, the only thing that they transplant was just like AB and DR, and I've seen patients 30 years, 40 years transplant doing well. Mm-hmm. So with this old technology, are we going to reduce transplantation or?
That's a great question. I see these. He works in the HLA lab like I do, and you see these false positive reactions all the time in the lab. And I often say, I wish I could just go back to serology. It'd be so much easier, because some of those patients do well. But for the ones that don't do well, I think we owe it to them to try to understand why they're in the disadvantaged group. Do I think we're providing too much information to physicians? I think that's a question for the physicians. But I think it's not that. It doesn't preclude us from learning the knowledge. I think it's more about educating and presenting that information. Like, I don't think I would want to throw a 25 gene panel at a physician. They'd be like, I mean, if I saw that, I wouldn't know what that was. But if you said to me, based on all these pieces of information, I think that this patient has a risk score of 7, and they know what 7 means, or we know what 7 means. I think that would probably be the more useful way of transmitting all this information. But underneath that single score goes decades and millions of dollars of understanding how we build that accurately for our patients.
Thank you.